Hello there. Welcome to Fundamentals of Biology, Lesson 4, where we're going to have a look at lipids, which is the second of our classes of biological molecule. Following on from the last session then, where we looked at carbohydrates, today we're going to have a look at lipids. I'm going to talk to you a little bit about lipids first, so we'll have a little introduction. And then I'm going to focus on a couple of types of lipids in particular. And the first one that we're going to look at are called triglycerides. So we'll have a look at those. And they're typically, we refer to them as fats and oils. So these will be ones that you're familiar with. We're going to look at how we actually make triglycerides or how they're made in nature through something called Ester linkages. And what we'll see is that we also see that these ester linkages are created by dehydration synthesis. So the same process that we saw in the last session with carbohydrates. We're then going to talk about the difference between saturated and unsaturated fats. We'll then go on to finish this section on lipids by looking at phospholipids, which are another type of lipid and really important for life because they are one of the main structural components of all cell membranes. That's the aim for this session then. Why are lipids essential for life then? What are they? What do they do? Well, if I asked you to tell me the first thing that pops into your mind, if I ask you about lipids, it's probably going to be fat. Or some of you might think oils like olive oil or something like that. But typically when we think of lipids, we think of fats. And that's absolutely right. But fats are just one type of lipid. When we start looking at them more closely, we can see that lipids are a really diverse family of molecules. And not only do they include fats and oils, but we also get the phospholipids, so important for cell membranes, but we also get waxes and also steroids. We can see there that there's a really diverse range of functions and types of molecule that we get in this family of lipids. So what are some of the things that they do then? Well, there's all kinds of important biological functions that lipids have. And we know about fats, and we'll talk more about them a bit in a minute. But fats, the primary role for those for fats is energy storage. So we talked in the last session about carbohydrates. And when we get a surplus of carbohydrates in our food, we can convert the simple sugars into glycogen and store it in our liver and muscles for later use. But any additional carbohydrates can be further transferred or transformed into fats. And we can then use these for long-term energy storage. But we also find lipids forming the structural components of cell membranes. So I mentioned phospholipids just now. But also cholesterol. So cholesterol is a steroid. It belongs to this group of lipids called steroids. And this also includes the sex hormones, so estrogen and testosterone. But we also get, you can see on the diagram at the bottom there, where we have 4 of the steroids, so testosterone, estrogen, cholesterol, and progesterone. But we also can include vitamin D in that. So vitamin D is formed by the UV light in sunlight hitting our skin, and then the cholesterol or some of the cholesterol in our skin cells is transformed into vitamin D. I think we typically think of proteins when we think of hormones, but we do get this group of lipids, so these steroids that are hormones as well, and they have incredibly powerful effects on our bodies. We have testosterone that is typically or produced in large quantities in males that lead to the development of secondary sexual characteristics in males and has a big role in sexual development and reproduction. We have the same thing in females with estrogen. And then cholesterol, which I mentioned just now, is an important precursor for those, and also vitamin D, but is also an important component of cell membranes. And we'll look at that in more detail in a later lesson when we look at cell membranes. We can just see there's a really, really diverse range of lipids and also a diverse range of functions that they have. And I didn't mention there actually the waxes. So we see waxes quite a lot in nature, particularly where organisms need to waterproof themselves. So we'll see a lot of plants with and insects with what we refer to as a waxy cuticle. And this is like a waterproof covering that helps to prevent water from sort of sitting on the surface of the leaf or sitting on the surface of the insect. And instead, it will sort of run straight off. If they're such a diverse group of molecules, then why do we include them together in the same family? What makes a lipid a lipid? Well, one of the main features that all lipids share is their low solubility in water. And what that means is they are what we refer to as hydrophobic. So hydro referring to water and phobic referring to sort of a dislike of or hating. So they are water hating. Lipids do not mix well with water. And if you've ever done the washing up and you've washed an oily frying pan or something like that, you'll notice that when the oils get into the water, they don't dissolve in the water. Instead, they sort of float to the top and you get these globules of oil mixing. And that's because lipids have a low solubility in water. They do not like to mix with it. Okay, so I want to start by looking at the triglycerides with you. So I said I wanted to look at two types of lipids in a bit more detail with you. So triglycerides and phospholipids. So let's start by having a look at the triglycerides, what they are and how they're put together. In animals, when there's an oversupply of dietary carbohydrate, the excess carbohydrate is converted or can be converted to triglycerol, which is fat. And this fat is stored in adipose tissue. And adipose cells, which make up this tissue, are almost completely filled with fat. And you can see in the diagram, the bottom right there, that this is a close-up view of adipose tissue. And you can see that this forms what's called the subcutaneous layer, which sits under the skin. And with these cells, you can see that each cell has a nucleus, and then the rest of them is filled with these fat droplets that are made up of triglycerides. Those triglycerides, that adipose tissue has a range of important functions within animals. So not only do we use them for energy storage just for ourselves, but other animals will produce them for hibernation, but also animals that undergo long migrations. So when you see migratory birds, for instance, they will spend time typically in warmer areas where they're able to access plenty of food, and then they will build up their fat reserves, and then those fat reserves will be able to power their migration. Similarly, with hibernation, if you look at something like grizzly bears that will undergo annual migrations, they will be able to spend the spring and summer months where they absolutely gorge themselves and put on as much weight as possible. And then that fat will actually see them through the period of hibernation. And by the time they emerge from hibernation the following spring, they will be really, really skinny, and then they will start the whole process over again. In fact, if you're interested, I believe it's in Canada somewhere where certain local authorities or local groups actually run a fattest bear competition. So they do an online thing where they award the bear that's put on the most weight during the summer, which is quite good fun. So yeah, so the build-up of these triglycerides then is really important for energy storage. But it also has other roles. So this adipose tissue, it's not just for storing energy, but it also provides insulation, and it also cushions the organs. Most animals will actually develop a small amount of fat around certain organs, like the kidneys and even the heart. This can actually provide some cushioning for them. But probably the biggest thing, or the biggest function, or the most important function behind energy storage that triglycerides have is insulation, particularly with certain groups of animals like marine mammals. So if you look at something like a humpback whale that is moving into really, really cold water to feed, like the Arctic or the Antarctic, well, there the water is close to 0 degrees Celsius. And it would be completely impossible for those whales to survive in that water for very long at all without a serious amount of insulation. And that insulation we know of as blubber. And that blubber is essentially this adipose tissue that's made up of triglycerides. And what that does is it puts a barrier between the insides of the whales, which need to be maintained at a similar body temperature to our own, so sort of in the mid to late 30 degrees Celsius, and the really cold water that's surrounding them. If they didn't have that adipose layer, that thick blubber layer, then the heat that from their bodies would just dissipate out into the water, because water is a fantastic conductor of heat. And it really wouldn't end well for the whales if they didn't have that blubber layer to insulate them. We can just see there then that some of the important roles of triglycerides. Yes, absolutely, it's important for energy storage, but also it has a role as for cushioning organs, but also it's incredibly important for insulation for animals that live in cold environments. And up in the top right hand corner here, you can see the molecular structure of a triglyceride. So what we're going to do next is we're going to examine that in a little bit more detail, and we'll talk about how we make it. Triglycerides then. When we look at all lipids, we see that they're made up of carbon, hydrogen and oxygen. And the most common type of lipid is the triglyceride. So that's what we're going to look at next. A triglyceride is made up of two main components. The first component, which we can see in the diagram here, is a molecule of glycerol, and glycerol is an alcohol. But you can see it's just made up of carbons, oxygens and hydrogens. We've got 3 carbons, and that's surrounded by hydrogens here. And then we have 3 of these things. And hopefully you recognize these from the last session. And if you can remember the last session, I talked to you about something called functional groups. And I showed you 4 functional groups, a carbonyl group, a hydroxyl group, a carboxylic acid group, and an amino group. And hopefully you'll be able to look at this and you'll be able to recognize that what we have here are 3 hydroxyl groups. And if you remember what I told you about functional groups, it's they have predictable chemical behavior, and that when we see them as part of a molecule, it often tells us that this is where we're going to get a chemical reaction occurring. So if we try and break the word down, so we're going to build a triglyceride or triglycerol. So tri refers to 3. The glyceride refers to the glycerol, and we've got 3 hydroxyl groups. So we're going to make a triglyceride. So hopefully you're starting to get the picture now. We're going to take 3 different molecules and add them onto these 3 hydroxyl groups. And the 3 things that we're going to add to them are called fatty acids. So that a triglyceride is formed when 3 fatty acid chains join on to each of the hydroxyl groups. And each time they do that, it's done through dehydration synthesis. So I said in the last session that we would see that process over and over again. Well, we're going to see it again here. So dehydration synthesis, the removing of a molecule of water and forming of covalent bonds to make a larger molecule. And when we do that, we're creating something called an ester link at each one of these hydroxyl groups. So in chemistry, an ester bond or an ester link is just a bond or link that's formed between an acid and an alcohol. The glycerol, that's our alcohol, and then we're going to take a fatty acid and put it one on there, and then a third one on there. So let's have a look at, let's walk through that and have a look at what it looks like. Here is our glycerol molecule, and we've got our 3 hydroxyl groups. What we're going to do is we're going to take 3 fatty acids. So don't be, I know that doesn't look particularly pretty when you look at it, but don't worry, it's quite a straightforward molecule. We get lots of different types of fatty acids, and they all have slightly different names, and they're all dependent on or what fatty acid you have is dependent on how many carbons you have in the chain and some other functions, but they come in different lengths and they're different types. But we're going to look at these 3 fatty acids here, and as you can see, they're all just long chains of carbons surrounded by hydrogen. So we would refer to these as hydrocarbons. They're a really important source of energy because there is a lot of energy stored up in the covalent bonds between these carbons and hydrogens. So as we start to split these apart and we start to remove the hydrogens from the carbons, and then we start to form new bonds, so when we start to make water and CO2 and things like that, then energy can be given off. So that's how these fatty acids, these hydrocarbons in this case here, are able to store lots of energy. So that's a fatty acid. We can see lots of carbons surrounded by hydrogens. This bit up here, hopefully you recognize this as one of the types of the 4 functional groups I showed you in the last session. Carbon double bonded to oxygen gives us a carbonyl group. Attach a hydroxyl group to that and you get a carboxylic acid group. So this is where we get the term fatty acid from. So this is the acid part of it. It's that carboxyl group or carboxylic acid group. And when we look at this now, we can see that we line this top fatty acid up to our glycerol molecule, and we've got a hydroxyl group here, and we've got a hydroxyl group there. So 2 oxygens and 2 hydrogens. Just like last week when we were making our polysaccharides and our disaccharides, we need an enzyme to facilitate the reaction here. So an enzyme will come along and it will attach to these molecules, and then it will start to break off molecules of water. So let's have a look at that. So in the red boxes here, we can see that an enzyme will break off hydrogen, another hydrogen and an oxygen. So that's H2O, same again here, and then the same again here. So for each triglyceride that is produced, how many molecules of water do we produce? I heard you all say that then, that was great. Yep, we produce 3 molecules of water. So we get a molecule of water here, a molecule of water from here, and then a molecule of water from here. The next thing that the enzyme will do is it will facilitate the making of a covalent bond between this oxygen and this carbon. Okay, so we're now going to join this carbon to this oxygen. We've removed the water molecule from there, and now we can just join that oxygen to that carbon. If we go back to our first session together, we can look at the principles that we talked about there. We know that oxygen, with its atomic number of 8, has 6 valence electrons, so it wants to make 2 additional bonds. Well, 1 additional bond goes to this carbon, and then the second additional bond will now go to this carbon. We know that carbon can make 4 additional bonds, has an atomic number of 6, so 2 electrons in the first shell, 4 in the second shell, so it wants to make 4 additional bonds. 2 go to the oxygen there in that carbonyl group, third one goes to the carbon next to it, and then its 4th covalent bond will be made with this oxygen over here. And it's the same down here and the same down here. So what we end up with is 3 fatty acid chains joining on to this glycerol molecule through dehydration synthesis. And every time we get a link now between this oxygen and this carbon, that is now called an ester link or an ester bond. And an ester link is a bond between an alcohol, so our glycerol, and an acid. In this case, it's our fatty acid. So we'll have 1 ester link here between the oxygen and the carbon. We'll have a second ester link between this oxygen and this carbon, and a third ester link between this oxygen and this carbon. Once that's happened, then we start to get something like this. So this is these are made up of slightly shorter fatty acid chains, but it's the same principle. We started off with a glycerol molecule, which we can see here with our 3 hydroxyl groups. Here is our glycerol molecule now after it's undergone the process of dehydration synthesis. So here's the oxygen. If we look at this oxygen here, we can look up now, and that's here. Here's the ester link, and this is where it attaches to the fatty acid. Same principle here. So if we keep following this oxygen, that oxygen is now here, and it's joined by dehydration synthesis to create this ester link between the fatty acid, and it's the same down here. Hopefully that's I say fairly straightforward. It's not the most straightforward thing to follow the first time you go through, but hopefully it wasn't too bad. The next thing that I want to mention was, if you look at the bottom of this diagram, we can see that this is a triglyceride and that it is what we refer to as a saturated triglyceride. So you're probably all familiar with the terms saturated and unsaturated fats. So what we can see here is an example of a saturated fat. And typically fats, and again, there are exceptions to this, but typically saturated fats come from animals or animal products. They are solid at room temperature. And so classic examples where you can think of some whilst you're watching this. So think of some examples of fats that are solid at room temperature and typically come from animal products. So hopefully you're thinking about things like butter, lard you might be familiar with if you do baking. So those are good examples. If you've ever cooked bacon in a frying pan, once you're cooking the bacon, everything sort of sizzling and liquid, and then you make your bacon sandwich, and then you come back an hour later and the frying pan looks very, very different. It's all solidified and it's full of a white fat. Well, those are saturated fats that have become solid at room temperature. And what makes a saturated fat saturated? It's all down to these fatty acid chains. So if we look at all three of these fatty acid chains, starting from the first carbon behind what was the carboxyl group, now it's just this carbonyl group here. So we'll start from this carbon here, the second one back. If we look at that carbon, we can see that it has 4 single covalent bonds. So this means that this carbon has a single covalent bond with that carbon, a single covalent bond with that carbon, and then the other 2 remaining bonds are made up of single bonds with 2 hydrogens. Come back to this one, exactly the same. Back to this one, exactly the same. What this means is, when we follow the structure of this saturated fatty acid around here, what we can see that it is full of hydrogens. There is nowhere else where we could put any more hydrogens. Each carbon has the maximum number of bonds filled that it possibly could have. So that's why we say it's saturated. It's saturated with these hydrogens. Now, this is important because when we get saturated fatty acids, the structure of them is very, very flat or very straight. These fatty acid chains are very, very straight. And what that means is they can pack together quite tightly. And that's why at room temperature, they tend to become solid. You can imagine them just sort of sitting on top of each other and packing together to form solid material. And that's basically what they do. And it's one of the reasons why it's often suggested that we need to avoid having too many saturated fats in our diet. Because for a long time, there have been a lot of suggestions that these saturated fats will actually solidify or clump together in our blood vessels, and they'll start to form plaques that actually narrow the diameter of our blood vessels, and it can harden our blood vessels. Now, there's been quite a lot of debate about that over recent years, and there's certainly some good arguments to suggest that saturated fats in our diet aren't the evil that we've been led to believe. But there are still a lot of dietary scientists that suggest that we need to moderate our saturated fat levels. My suggestion would be to you, if you are interested in nutrition and you like that kind of thing, then certainly do some research into it. It's not the black and white argument or topic that it was, say, 10 or 20 years ago, where saturated fat was bad, we must cut it out at all costs and replace it with unsaturated fats. Now that we're moving into slightly more of a grey area, and there are good arguments for saturated fats in our diet, and there are good arguments against it. So I'm a firm believer personally in moderation. So I don't think saturated fats are the worst thing in the world for us, but I think it's safe to say that if we are eating nothing but fish and chips and McDonald's every day of the week for our sustenance and our nutrition, probably bad things are going to happen to us at some point. So it's one of those things in moderation, in a balanced and varied diet that incorporates fruits and vegetables. Then some saturated fat isn't necessarily going to cause any problem. But I am no dietitian. I would leave it up to you to do your own research on that. But that's what saturated fats are anyway. They have these nice straight fatty acid chains because all of the carbons have single bonds and they are saturated with hydrogen. If we move on to the next slide, then what we can see here is a comparison between a saturated single fatty acid and an unsaturated fatty acid. So in the fatty acid chains, we can see here the carbon atoms may have single bonds between them. And if this is the case, which we can see here, that lipid will be saturated. These are usually solid at room temperature and are called fats. And like I said, we typically get those from animal products. Just be aware that it's not, like I said before, there's exceptions to most rules, and we do find a lot of saturated fats in certain plants like palm oil and things like that. But and coconut oil has some saturated fat in as well. But typically we get saturated fats from animals. However, we can also get unsaturated fats that we can see represented in the diagram here. What we can see here, if one or more bonds between the carbon atoms in the fatty acid chain are double bonds, then we would class the lipid as being unsaturated. And unsaturated fats are typically liquid at room temperature, and we refer to them as oils. And these will typically come from vegetables. So you can think of probably quite a lot of examples off the top of your head. So if you think of a fat, that is an oil, so liquid at room temperature, typically comes from plants. Well, you're looking at things like olive oil, sunflower oil, rapeseed oil, things like that. But just be aware that, as I said earlier, there are exceptions to most rules, and we also find a lot of unsaturated fat in fish. So although fish are animals, they do contain an awful lot of unsaturated fat. And there are good reasons for that we can talk about at a later point. So if we look at the structure of an unsaturated fatty acid then that we can see here, we can see at first the structure is the same as a saturated fat. So we've got these carbons that have these single bonds. They're all full or saturated with hydrogens until we get to this part of the molecule where we can see between these two carbons there is a double covalent bond. So if you remember from our first session, a double covalent bond is the sharing of two pairs of electrons. Where we have a double covalent bond between carbons of the fatty acid chain, what that means is, in theory, if you could break that bond, or you could break one of those bonds, you could add another hydrogen here to this carbon, and then you could add another hydrogen here to this carbon. So there's room for two more hydrogens in this fatty acid chain. That means it is not saturated with hydrogen, so it is unsaturated. That has a big impact on the structure of the fatty acid chain, and you can see it in the diagram. As you come along, just like a saturated fat, where you get the single covalent bonds, the structure is straight. But where you get a double bond, you get a kink in the chain. It sort of moves off at an angle. And what that means is now that we have this kink and the fatty acids are no longer straight, it means they will not and do not pack together tightly like the saturated fats do. So at room temperature, that means that they are oils, they're liquid. And it's believed that the argument that I mentioned just now about the levels of saturated and unsaturated fats in our diet, this is the reason why unsaturated fats are considered to be better for us. Because they remain more liquid, they won't pack together tightly at room temperature, and in theory they shouldn't do that inside of our bodies either. So that's the argument for unsaturated fats. And there's also the added argument that we get unsaturated fats largely from plants. And when we consume plants, we get a lot of other additional benefits like certain vitamins and minerals. What we can see here then is what's often referred to, or what would be referred to, as a monounsaturated fat. So it's mono meaning one. It means that there is one double bond between these two carbons. And that means that there's one kink in the chain. But you might have heard of something called polyunsaturated fats. And a polyunsaturated fat would be a fatty acid that contains two or more double bonds. So the more double bonds you get in a fatty acid chain, the kinkier it is, basically. So every time you get a double bond, you get a kink. So the more double bonds, the more kinks there are in the chains, which means, in theory, the more liquid, the more fluid they remain. So in theory, the healthier they are for us. That's the difference between saturated and unsaturated fats then. It comes down to the molecular structure and how the differences in the molecular structure will cause differences in the way they behave. So saturated fats are solid at room temperature, and we typically get them from animals, and we refer to them as fats. Unsaturated fats are typically liquid at room temperature. We get them largely from plants, and we refer to them as oils. That kink in the tail basically means that they won't pack together tightly. This can cause a slight problem when it comes to food manufacturing. If you're manufacturing something like biscuits, for instance, you want a product that is solid at room temperature. Nobody wants to take a biscuit out of a packet and for it to be liquid and to slop apart in your hands. You want to be using, ideally, saturated fats in the manufacture of your biscuits. But if you are a manufacturer of biscuits, you've set up your own biscuit company, the problem that you've got is, as we've already discussed, to get saturated fats, you typically get them from animals. And if you want to grow animals to get fats to make your biscuits, it's really expensive. Animals require a lot of money. They need room for housing. They need feeding. They need veterinary care. They then need slaughtering and processing. So there's quite a lot of work and expense involved. It would be much easier and much better if you could grow plants, basically, and that would ultimately lead you to working with unsaturated fats. Now, that obviously causes the problem that you get oils from these plants and not the saturated fats that you need. There's two ways around this. One, you can grow a type of plant that produces a decent amount of saturated fat. The number one for that being palm oil. So that's why we're starting to see, or we have been seeing a huge increase in the amount of palm plantations around the world. So we can get palm oil, which contains the saturated fats that we need for making all manner of products. Another thing that you can do, and this was really popular for a few decades, from about the 1970s through to sort of the early 2000s, and this was taking our unsaturated fats, and we would get them from plants like corn and soy, and then you can actually use chemistry, or witchcraft as I prefer to think of it, and transform them. And what they would actually do is you would take the unsaturated fats like this, and then you would put the oils into big vats, and those vats would be heated and put under pressure, and then you start to bubble hydrogen up through the oils. And the combination of the heat and the pressure and the hydrogen would ultimately lead to these bonds, these double bonds between the carbons breaking, and then the addition or the changing of these bonds over here, and the addition of more hydrogens. And that would enable that would change the structure, removing these kinks and making the fatty acid chains straighter. So you suddenly have a product that you've gotten from unsaturated fats, and you've basically got something that behaves more like a saturated fat. And these are what are referred to as trans fats or trans fatty acids. And the process is called hydrogenation. So these are probably terms that you're familiar with. Now, trans fats have been pretty widely proven now to be worse for us although it's difficult to prove exactly why they're worse for us, but they do seem to be linked with higher incidences of heart disease and obesity. And like I said, it's as far as I'm aware at the moment, there's no conclusive evidence of why that is. But it got so bad that a lot of countries have actually now banned the use of trans fats in food. And in the UK at the moment, although they aren't banned, I think it was back in 2012, there was an agreement made between European countries and food manufacturers that they would greatly reduce the use of trans fats in our food. So that's quite an interesting thing about, and we can see how our real lives are impacted by biology and chemistry, in the food that we eat and other products that we consume. A lot of this comes down to moderation. I think that the healthiest way that we can go is to eat a balanced and varied diet. It's okay to eat unsaturated fats. It's okay to eat saturated fats. And it's about balancing it out with healthier options and exercise as well. Okay, so hopefully that made sense anyway, with the difference between saturated and unsaturated fats. The last bit that I wanted to talk to you about in this section is a different type of lipid, and it's one called phospholipids. I mentioned earlier on how phospholipids are an important component of our cell membranes. So let's have a little look at them, and we will talk about these in a bit more detail in a lesson that I'll do with you on cell membranes. So this is more of just a gentle introduction to them. So phospholipids then are important components of our cell membranes. If you've not studied any biology before and you're not familiar with cells, well, every form of life on the planet is made up of cells, from single individual celled organisms like certain bacteria and things like that, right through to complex multicellular organisms such as ourselves, where we're made up of anywhere between 30 and 100 trillion individual cells. But every single cell of every single living organism on the planet is made up of phospholipids, and there's also a lot of proteins in there, but phospholipids are one of the main components of cell membranes. And phospholipids are quite interesting because they share a lot of similarities with the triglycerides that we just looked at. So like fats, and so the triglycerides, phospholipid molecules contain fatty acid tails linked to a glycerol molecule. So if we look at the diagram here on the right hand side, what we can see is this section here that I'm highlighting is a glycerol molecule. And then coming off of that glycerol molecule, we have two fatty acids. So we can see a saturated fatty acid here that's nice and straight. We can see an unsaturated fatty acid here. You can see the double lines here showing us that it's a double covalent bond between the carbons there. So that one has a kink in it. So we have two fatty acid chains. Now, obviously, the key difference here is there isn't a third fatty acid chain that would give us a triglyceride. Instead, that third fatty acid chain is replaced with a phosphate group. So you can see up here, the phosphate group is a phosphorus surrounded by oxygen. And this little R up here just denotes that phospholipids will often have other molecules sort of attached up here, but that bit isn't important for us right now. The main bit here is that a phospholipid is made up of a glycerol, two fatty acids, and instead of the third fatty acid that would give us a triglyceride, we have a phosphate group attached to the glycerol molecule instead. Now, that might not seem very important, but it really is, because it changes the behavior of phospholipids completely from triglycerides. And that's because I mentioned right at the beginning of this that one of the behaviors that, or the main behavior that all lipids share, and the thing that groups them all together, is their low solubility in water. They are all water hating or hydrophobic. But phospholipids are a bit of an oddity in the sense that part of the molecule, so the part of the molecule here that contains the fatty acid tails, this is hydrophobic, as with most lipids. So we can see here, we often refer to them as the hydrophobic tails. But the phosphate group that we have up here, because of all of those oxygens, this part of the molecule is polar. And because it's polar, it can make hydrogen bonds and bonds with water. So we would refer to this part of the molecule, the phosphate containing section, as being hydrophilic. So we often refer to this as the hydrophilic head. That's why you can see this sort of surrounded by this red ball, and then you've got the tails here. So what we end up with is a molecule that is half hydrophilic, so water loving, and then half hydrophobic, water hating. And this is what we call amphipathic. It's a molecule that is has both hydrophobic and hydrophilic properties to it. So we can see here, this makes the molecule ambivalent with regard to water. Part of it will mix happily with water, part of it hates water and tries to get away from it. So part of it is polar and mixes with water. That's this part, the phosphate containing section. While part of it is non-polar, so there's no electrical charge on it, and it is excluded from water. It will not mix with water, and that's the hydrophobic tails. So why is that important? Well, this is where we believe we got the original cells from. So when life began originally, obviously it's impossible to prove at the moment, but it's believed that the very, very earliest forms of life possibly began as some simple chemical reactions occurring on a rock somewhere. And then at some point, we had some phospholipids appear. And when you add phospholipids to water, they will tend to form a couple of main shapes. And one of those shapes would have enabled our chemical reactions of life to occur independently of the rest of the world. And I'll go through this and I'll explain what I mean. So when phospholipids are mixed with water, they will spontaneously arrange themselves. And they'll do this because the hydrophobic regions, so those fatty acid tails, will try really hard to find ways to get themselves away from water. The hydrophobic parts they hate water, so they don't want to be in contact with it. Whereas the hydrophilic regions, those phosphate heads, they will try and interact with water. They will happily make bonds with water. So if we look at the diagram here, we can see what can happen. If you were to take some olive oil and pop it into a glass of water, what you would tend to find is you would get this kind of appearance. You would get this sort of slick on top as the oil kind of coagulate. They sort of form together, and they're at the surface trying desperately to get away from the water. They'll actually sit on top of it. If you add phospholipids to water, they can do something similar, so they can form a slick on top, but these red heads, these hydrophilic phosphate heads, will actually make bonds with water, so they'll actually be quite happy. And then you'll have these hydrophobic tails sort of poking out that will give you that sort of look of an oil slick. However, when they're in water, they can also form spheres. So they will form two types of sphere. The first one looks like this. It's a monolayer bubble. So the hydrophobic tails will all point inward. So if you could, although we're looking at circles here, if you could imagine that this is actually like a football. So it's actually like a sphere. And pointing inwards, you will have these hydrophobic tails. And they will exclude all water from the inside. So there's no water inside. These hydrophobic tails are really happy now. They're just pointing in towards an oil droplet. They're away from the water. The hydrophilic heads on the outside are happy because they'll just mix with the water that they're in. But that's not of much use to us, because for the chemical reactions of life to occur, they need water. But because we don't have any water inside here, you're not going to get any chemical reactions that enable life. Phospholipids will also form something called a bilayer, a phospholipid bilayer. And you can see that here. So imagine again, this is a football, not just a circle. What we can see is that we get two layers of phospholipids where the hydrophobic tails face each other, and then you get a layer of hydrophilic heads pointing inside the football, and then another layer of hydrophilic heads pointing out of the football. This is important because this means that the this sphere or this bubble can exist happily in water because the hydrophilic heads out here will happily bind or bond with water. But it also means that inside of the bubble, inside of the sphere, you can also have water because you've got these hydrophilic heads pointing inside the sphere. So you can get water in here. And what this means now, you can have all the chemical reactions that are required for life to exist happening inside this bubble where they are protected. They aren't being interfered with from anything that's going on outside. So you've got they've got their own little environment in here where they can go about their business unencumbered or uninterrupted by the outside world. And this is where we believe we got our very first proto cells from, our very first early cells. You get this separation of the inside or the intracellular world. So the inside of the very first cells. So that's separated now from the extracellular world, the world outside of the cell. So just that nature of phospholipids having a hydrophilic head and hydrophobic tails enabled them to form these spheres that contained water, and that enabled the very early life to sort of exist and then develop and evolve in a protected environment away from the outside. And that's why we find that today phospholipid bilayers form the cell membranes of almost all forms of life, from the simplest single-celled bacterium right up to blue whales and humans and oak trees. We're all made of cells, and our cells are made up largely of phospholipid bilayers. And we'll look at that in more detail when we talk about cell membranes. But hopefully that made sense about phospholipids. So that brings us to the end of our little section on lipids then. So let's just do some quick recap questions. We'll pause this in a minute and give you a chance to answer them, but let me quickly go through the questions. So number one, which reaction links fatty acids to glycerol? So what type of reaction is required for that? Number two, name 3 examples of lipid steroids. Number 3, what is the term for compounds that do not mix with water? And then number 4, the lipids that form the main structural component of cell membranes are called what? Okay, so we'll give that a minute and then we'll come back and we'll go through the answers with you. Okay, so welcome back. Hopefully you had a chance to answer all of those questions. Let's go through the answers. So number 1, which reaction links fatty acids to glycerol? Well, the answer is dehydration synthesis. I imagine some of you might have put ester bonds or ester linkages there. Now that's not the name of the reaction, that's the name of the bond that is formed as a result of the reaction. So the actual process of joining the fatty acids to glycerol is dehydration synthesis. And in the form of triglycerides, that link then, or that bond that's made, is called an ester bond. When we looked at carbohydrates in the last session, that was called a glycosidic bond. So what you'll notice is that for each of our 4 classes of biological molecules, so carbohydrates, lipids, nucleic acids, and proteins, each will have their own specific links or bonds that make them up. So carbohydrates was glycosidic, and with our triglycerides, it's an ester bond. Okay, number 2, name 3 examples of lipid steroids. The 3 main ones that I gave you were cholesterol, estrogen, and testosterone, but you could also have gone for progesterone or vitamin D. So any of those 5 would have been great. Question number 3, what is the term for compounds that do not mix with water? That's hydrophobic. And what's the opposite of hydrophobic? It's hydrophilic. So water hating and water loving. And then number 4, the lipids that form the main structural components of cell membranes. Well, what are they called? They're called phospholipids. So hopefully you got that one as we just did it. Okay, so that's just hopefully a fairly gentle introduction to lipids. So we've looked at of what lipids are and briefly mentioned the different types that we get. Then we focused on triglycerides, and we looked at how we make triglycerides from molecules of glycerol and fatty acids through dehydration synthesis to make ester linkages. And then we went on to look at saturated and unsaturated fats. So obviously the difference being whether or not we have double carbon bonds in the fatty acid chains. And then we just finished off by having a quick look at phospholipids and how their structures differ, or their structure differs from triglycerides, and also a little bit on why we believe that they enabled the formation of life by providing or producing those early proto cells. Okay, so hopefully that was interesting and useful. Thanks very much. I'll see you in the next session where we'll be looking at nucleic acids.